Awesome. Yeah. So thank you so much. So yeah, as you can tell from the kind of vague title, I am in the very early stages of the PhD. So I think my purpose today is going to be kind of laying out some of my methodologies, and what I hope is going to be a useful methodological approach in dealing with particularly large scientific collections, like those at Kew, which are obviously very, very rich resources of information about the actors that constructed them, and how these actors may have interacted with one another. So my paper's really focusing on examining the interplay of science and globalization through the context of 19th century British plant specimen exchange. So I focus mostly on the Royal Botanic Gardens Kew as a key case study, applying social network analysis as an exploratory lens that illuminates the often overlooked contributions of local collectors, laborers, and intermediaries in the transcontinental movement, plants and knowledge. So the Kew Herbarium housed plant specimens acquired through many colonial networks, representing really complex relationships and power dynamics. Yet historical narratives often privilege figures like William Hooker, his son Joseph Hooker, and Joseph's son-in-law William Thiselton-Dyer, whose combined directorships from 1841 to 1905 marked a period of significant expansion and collecting activity at Kew. But by decentering these figures, I hope to map the broader networks of individuals and institutions underpinning botanical exchange. So a focus on 19th century Australian plant collecting networks underscores the interdependence of colonial actors who both navigated and actively contributed to evolving nomenclatural and classificatory systems. So the first half of my talk will be a bit more methodological, and then the second half will be introducing some of the actors I plan to focus on during my PhD project. So it's a bit of a teaser trailer, I hope, for future talks. I hope you're left wanting a bit more. But just as a brief introduction to what social network analysis actually is. So it's a method of really studying interactions and relationships between entities. And these entities can be anything, right? They can help identify key contributors, bridges between groups, and influential figures in a network. And in herbarium specimens, social network analysis can reveal connections between collectors, institutions, and even plant species or families. So I'm interested in, at a further stage, looking at how collector communities aggregate around specific plant families, for example. But some key concepts in social network analysis are that you have these nodes. In my case, they represent individual plant collectors, and then edges, which represent the relationships between those collectors. So the edges I also assign weights to, and this weight just corresponds to the number of specimens a given pair of individuals have collected together, or in which they're mentioned on the same specimen label, which, as I'll get to in a little bit, can mean a few different things. So there are a few really important metrics in understanding networks. So degree centrality is the most common, and this just measures the number of direct links a node has to other individuals in their social network. So a high degree centrality will likely mean that a collector, shares specimens widely and works with a variety of collaborators. So in this example, a botanist who collects specimens with 5 other collectors has a higher degree centrality than one collecting with 2. I also look a lot at betweenness centrality as a way to complicate degree. So betweenness measures how often a node acts as a bridge between different groups. So high betweenness centrality means that a collector links otherwise unconnected groups, as we can see in the example at the center here at the bottom. So a collector working in a remote region, sending specimens to 2 otherwise unconnected botanic garden directors would be a great example of a node with high betweenness centrality. And finally, I look at eigenvector centrality, which is a measure of the importance of an individual actor. Obviously, that's a bit tricky, but it's a useful proxy I've found so far in considering both the number and the importance of connections a single actor has. So high eigenvector centrality means being linked to many influential collectors, or maybe a few very influential collectors. So a researcher working with a few well-connected individuals at Kew, for example, may have a high eigenvector centrality, despite having fewer direct connections than someone collaborating prolifically at a colonial botanic garden. And there's a lot of limitations to this approach that I kind of wanted to acknowledge at the start, and then I'll talk a little bit more about how I'm working through addressing them as part of my project. So as with any historical archive or records, there's going to be missing data. There's going to be a bias towards well-known collectors that are much better documented. Well, I think because we see a lot of names appearing on these specimens that haven't had their correspondence preserved, that don't necessarily produce a lot of publications, that don't appear in other literature, this does kind of fill in a gap, and that's what I'm hoping to show today. There's also an issue of data standardization. Different herbariums use different record keeping practices. So I'm going to talk through some strategies for getting all of these different ways of keeping track of collectors and data about specimens into a more interoperable format. There's a lot of great resources focused on making that possible today, as well as disambiguating names. So there's many duplicate names, misspellings, and this can really skew social network analysis because you'll have 10 nodes that are actually just representing one individual. I'm also experimenting with looking at how networks change over time. The methods I'm using right now only provide a snapshot, but I think it's really important to start looking at how we can observe the evolution of networks at Kew over time. And that's definitely going to be a future stage of my research. Also, as I mentioned, with degree centrality, especially, but with all of the metrics that I'm looking at, high centrality doesn't necessarily equal, high scientific output or value. It's really just a quantitative metric that I'm using to help me explore this really significant collection. But just to think about what herbarium specimens can actually tell us, quite a lot, really. There's a ton of information about places, habitats, people, the plants themselves. So they're used by scientists for a variety of applications, from studying the impacts of biodiversity loss and anthropogenic climate change, to broader studies on changes in plant flowering time. So a huge range of applications, as well as historical value. And I'm specifically interested in plant specimens in which multiple collectors are listed on the label. So this can help establish a close degree of social connection. Sometimes this can also be problematic as names are added later. So this is where disambiguation and data standardization are really helpful in establishing that there is a verifiable link between two individuals listed on a given specimen label. And for those of you that haven't worked with herbarium specimens before, I just have a couple examples of labels pulled up here. Um, so in terms of kind of the push to standardize data, that's made it really useful for social network analysis. There's the Darwin Core format. So the Global Biodiversity Information Facility has emerged as one of the key global repositories for biological specimen data and biodiversity information. And they use a Darwin Core standard, which includes some really useful standard metadata fields. So during the process of digitizing herbarium specimens, this is how the data is captured and put into a more interoperable, machine readable format that I can then use for fun stuff like social network analysis. So I'm focusing mostly on the recorded by field. So this is who collected the specimen. Oftentimes there will be multiple names listed there. And that usually means that they either collected in the field together or communicated about the specimen, sending it perhaps to one another for identification. There's a separate identified by field that I'm less focused on right now, but I think could potentially be useful later in understanding institutional affiliations and understanding how institutions are kind of deciding what gets to be a specimen or what gets to be a new species, for example. There's also a ton of information about dates, locations, taxonomic details. So it's a really, really rich metadata infrastructure that I'm trying to take advantage of as part of my project. There's also a ton of ways to disambiguate names. And I mentioned that as one of the key challenges that I've run up against. Because many collectors can appear under several different names or spelling, share similar names, and this makes disambiguation really difficult. There's been a ton of really fantastic crowdsourcing efforts to help out with this. So Bionomia is one of the main platforms that I'm relying on for disambiguating individual collector names. In this example, I have Australian collector that has 6 known pseudonyms. So it's really important to be able to aggregate these under a single unique identity. And that's represented by the Wikidata QID. So we can see here that all of the possible names are now listed under a single numerical ID, which is what I'm going to be using to help disambiguate individuals in my networks. Okay, so now I've kind of gone through, I think, some of the more boring bits. I'm going to spend the second half of my presentation looking at some of my case studies. So in my initial explorations of this as a way to explore herbaria, I took the 7 largest Australian herbaria from the Australian Virtual Herbarium database on GBIF and generated this network. So I thought this was a particularly interesting approach for understanding institutional connections through networks of co-collection. So I tried to represent a lot of things in this graph, so I think it can be a bit confusing. I'm still trying to figure out how to best navigate, I think, presenting information in this format. But we can see here that edge color corresponds to the herbarium in which the majority of specimens between an individual pair are stored. So I have a color key here. We can see it's skewed very dramatically towards the New South Wales herbarium, but there's also some pretty good representation from Melbourne in terms of who was co-collecting at the time. And the width represents the volume of specimens between a given pair. So a thicker line means they collected more specimens together. And then node color demonstrates betweenness centrality, while node size shows higher or lower eigenvector centrality. So a large darkly colored node has a higher betweenness and eigenvector centrality, and is therefore more likely to be important within the context of this network. So in this initial analysis, I found that a couple moss collectors were actually the most social and the most interesting to take a closer look at in this giant component of the graph. And you might notice it's actually a fairly decentralized kind of network if we go back here. Like there's a lot of people working in pairs or small clusters around Australia in the 19th century, and not a lot really forming denser collecting communities. But we can see here that the individuals with higher betweenness are actually moss collectors. So William Walter Watts, who was a reverend, and William Forsyth, who worked with the Herbarium of New South Wales. And they actually outrank a lot of really more established figures in colonial Australian botany, like Ferdinand von Mueller, who was the director of the Royal Botanic Gardens, Melbourne, or Frederick Bailey, who was the acting director of the Queensland Museum and Botanic Gardens, and even Joseph Maiden at the center here, who was the director of the New South Wales Herbarium and Royal Botanic Gardens in Sydney. So of course, this is kind of expected. Colonial garden directors, weren't always doing the bulk of collecting. But this is, I think, exactly why social network analysis is quite useful in recentering garden staff and independent collectors, like Watts, who was a missionary, as go-betweens for botanical knowledge exchange. So the role of these intermediaries was really particularly important in a colony as geographically remote as Australia, with fewer professional opportunities available in botany and horticulture. So I've also highlighted an interesting case here with Ernst Betsch, who was a botanical collector and assistant at the Sydney Botanic Gardens. So he really emerges as a central figure in linking the two herbaria in Victoria and New South Wales. He's a really key proponent of Ferdinand von Mueller's version of the natural system for plant classification, which was met with a lot of resistance by Joseph Hooker and George Bentham at Kew, as well as William Thiselton-Dyer, who was director at the time, who were trying to standardize their own system of natural plant classification. So Betsch was a really key figure upon looking deeply into his correspondence in ensuring that both the two major herbarium in Australia were on the same page in adopting Mueller's system. And that made Australia the only colony in which Mueller's system was adopted in about 50% of publications and flora towards the end of the 19th century. So he did have a really significant impact in ensuring that Australia was kind of united behind their version of the natural system as opposed to Kew's version. And eventually it petered out. And I think there's a really interesting history I want to explore there in one of my chapters. But it does kind of show how Australian colonial botanists were diverging from the central authority of Kew on some taxonomic matters, which I think is quite interesting. And as a little bit of background on the Kew Herbarium itself, so it was formally founded in 1841, and the Kew Herbarium is one of the world's largest and most biodiverse plant collections, with over 7 million specimens. And since 2021, the Kew digitization project aims to make all specimens available digitally via the Global Biodiversity Information Facility by 2026. So my PhD research is really taking place in the context of this much larger digitization project, which has been ongoing for several years. So this is my preliminary network. A bit of a mess at first glance, but it takes approximately 10,000 individual collectors. And this is before proper data cleaning and disambiguation. So the actual number will probably be quite a bit smaller. But it's also an incomplete data set. So this is really just my first attempt at getting a snapshot of the Kew Herbarium and using that to guide some of my readings of the really extensive Kew archives. But as you can see, it ended up being about 10,000 individuals, taken from around 34,000 instances of co-collection and specimens, and the total 500,000 specimens that have been digitized so far from the period that I'm interested in, which is during the first three tenures of the directors at Kew, so the first three directorships. And importantly, I use a community detection algorithm here to color in loosely, and I think it works pretty well, loosely connecting communities that I might be interested in exploring further. So I also use node size here to correspond with eigenvector or the relative connectedness to other important individuals within the network. And that was quite helpful in kind of getting a few key characters to jump out at me. But I hope that this helps to complicate the idea of Kew as this kind of botanizing imperial octopus. You can get a much broader sense, I think, of the importance of local collectors, and of, as I'll talk about in a moment, powerful patrons of botany like Ellen and Wilmot, who were funding a lot of plant hunting expeditions that were feeding plant specimens into Kew. So I think Ellen Ann Wilmot has emerged. This kind of jumped out. She has the highest eigenvector and betweenness score of the graph that I generated. So she really jumped out as an influential figure. And she was a huge patron of plant hunting expeditions. Her funding facilitated plant collecting across the British Empire, including Central Asia, China. She funded Ernst Wilson's expeditions to China several times, and this really shaped the introduction of many species into British horticulture. Uh, and she was also one of the first 15 female fellows of the Linnaean Society. So she was really well situated within elite scientific and institutional circles. But I think her contributions have often been overlooked. And I think this is a really interesting way of kind of seeing where she fits within the much broader landscape of plant collecting at the time. And indeed, like showing her incredible significance, I think, to contributing to Kew. So networking her specimens kind of highlights the importance of her work in funding both Kew-affiliated plant hunters around the world, but also creating a very impressive herbarium of her own, which she later donated. And I think a lot of these specimens are represented because of that large bequest, which interestingly occurred when she was on the verge of bankruptcy. So she was very, very interested in kind of, I think, keeping her social capital and influence within the botanical elite alive, even while going through financial difficulties. So again, hopefully this will become another chapter. I think I'm running a bit low on time, so I'll just kind of skim through my last case study here, which I think is one of the more interesting ones, but unfortunately one that I have not had a lot of time to look at. But if we take a look at James Ramsay Drummond, so this kind of small cluster at the bottom of the graph here, he began a 30-year career in the Indian Civil Service in 1874, and then went on to collect quite prolifically with a lot of local Indian collectors. And that's something that's quite notable because the purple cluster in the center of the graph actually represents one of the directors of the Calcutta Botanic Gardens. But as we can see, this cluster is quite separate from that. So it's this kind of community of mostly local Indian collectors working with this colonial official, again, quite detached from the Calcutta Royal Botanic Gardens in India. So I think this presents an interesting case study for showing how we can really draw out some interesting communities of local collectors and situate them within the broader landscape of plant collecting at the time. Yeah, just to quickly wrap up, so I think that combining archival research with social network analysis really helps to explore transcontinental patterns of plant collecting, and leveraging specimen label data to visualize connections between collectors is a really useful way of doing that. So social network analysis provides a guided means of reading, not an objective one. And conversely, I think close reading of the archive can help inform the development of more nuanced and focused quantitative approaches in the history of science. So by foregrounding the contributions of local collectors, horticulturalists, and other intermediaries, social network analysis helps to complicate the perception of Kew as the sole locus of botanical progress and illustrates how the digitization of scientific plant specimens yields a really novel lens for surveying Kew's colonial archives. So I'll wrap up there. But thank you very much. I'm looking forward to some questions, hopefully.